Welcome, everyone. Today we're diving into the heart of immunohematology, genetics. You might wonder why we need to be geneticists as well as lab scientists. It's because every single red cell antigen we test for in the laboratory is an inherited trait. From the ABO on your ID badge to the rare Cal or Duffy markers, your genotype, which is the DNA you inherited, dictates your phenotype, the antigens we actually see reacting under the microscope. Understanding this code of life is what makes safe blood transfusion possible. Now let's talk about genes. Gene is a unit of inheritance that encodes a particular protein, and it's the basic unit of inheritance of a trait. When we talk about a DNA, it is where the genetic information are carried through our chromosomes. So proteins are coming from the genes. So in which there are codons that could code for a particular amino acid. When amino acids are grouped together, then protein is synthesized. Genes located in our DNA. The DNA is a double-stranded helix which contains the codons that are responsible for the coding of proteins. DNA, it is a coiled structure which is found in our chromosome. And let's review our chromosome. Our chromosome has 23 pairs, 1 pair for sex chromosomes, and 22 pairs of autosomes. Chromosomes are found in the nucleus of our cells, which is responsible for the control of metabolic processes inside the cells. So again, when we group cells together, they become tissues, and tissues are grouped together, they become organs, and organs when grouped together, they become organ systems, and organ systems grouped together now becomes the human body. So just a quick recap. Inside our DNA, we have nitrogenous bases. We have your adenine, thymine, guanine, and your cytosine. So 3 consecutive nitrogenous bases contributes or constitutes a codon that carries a particular amino acid. So amino acids are linked by peptide bonds in order to form a protein. So in the laboratory, we perform serologic testing to find the phenotype. Phenotype is the physical expression of your genes. So for instance, if your cells agglutinate with anti-A, your phenotype is group A. But does it tell us your genotype? Not necessarily. You could be AA, which is homozygous, or AO, which is heterozygous. Because the O gene is an amorph, meaning it's silent and produces no detectable antigen. It hides behind the A or B antigen. So in order to find the true genotype, we'd need family studies or molecular DNA testing. Again, let's just review. When we talk about phenotype, it is the outward expression of your genes. So on the red blood cells, serologically, demonstrable antigens, it constitutes the phenotype, except those sugar sites that are determined by transferases. So the antigens present on all our red blood cells are expressed as phenotype. While on the other hand, genotype is an individual's actual genetic makeup. So it's the genotype of the organism that controls what antigens may be expressed on the red cell. So let's say, for instance, if it is impossible to have a clear picture of the red cell antigens present on the red cells of a donor or recipient, or if an antibody screening give ambiguous results, genotyping the donor or recipient can let medical technologies know what antigens may be present on the red cells. So I have here a table of our ABO blood groups with their respective antigens present and as well as the antibodies they produced. So we have here an example. This is somewhat like a Punnett square. So let's talk about the left parents. So the parents here are classified to have phenotypes A and B, but genotypically they are heterozygous because a pair a parent or parent A as AO and B parent or parent B as BO. So if gene combination takes place to determine the genotype of the offspring, there could be a possibility that the offspring could have either AB, AO, BO, or OO. OO as a homozygous blood genotype, and then while the rest are heterozygous. So again, let's just recap that the phenotype is based on antigens present on the red cells, and it is the byproduct of the genetic makeup of your red cells. So if parents have A and B genes, then both antigens will be expressed, and then offspring will be typed as AB. Now, we have here a Punnett square. It is a visual tool used to display the frequencies of different genotypes and phenotypes among the offspring of a specific genetic cross. So in immunohematology, it serves as a probability map to predict the blood groups of children based on the known or inferred genotypes of their parents. So the basic mechanism of a Punnett square, it operates on Mendel's law of independent segregation, which states that each parent possesses a pair of genes for a trait. And these genes segregate so that only one gene from each parent is passed down to the child. So by placing the alleles of one parent across the top and the other parent down the side, you can visualize all possible combinations in the resulting grid. So let's say for instance, in blood banking, it is really common to use Punnett squares to explain how parents with certain phenotypes can have children with entirely different ones. So I'll give you a scenario. So let's say a mother with group A blood and a father with group B blood. So the genotypes, if both parents are heterozygous, the mother is AO and then the father is BO, the Punnett square reveals 4 possible outcomes for their children. So the offspring could be either group AB, group A, group B, or group O. So the result, there is a 25% probability for each blood group. So this explains why two parents who do not appear to be group O can still have a group O child. So I have here another example. Let me give an example in the RH blood group system. So Punnett squares are used to illustrate the probability of an infant being a D positive or a D negative, so which is critical in managing cases of hemolytic disease of the fetus and newborn. So let me give you a scenario again. So a mother is RH negative, which contains homozygous small d antigens, and the father is RH positive but heterozygous. So the father could have the big D and then the small d antigen. So the mother can only contribute a small d allele, while the father can contribute either a big D or a small d allele. So the result could show 50% chance of the child having the big D and then the small d, which will become RH positive, and also a 50% chance that the child will be small d homozygous, which will result now into an RH negative blood type. So why does Punnett square matters in the laboratory? So while modern laboratories often use molecular DNA-based assays to determine a true genotype, the Punnett square remains the standard for interference, so predicting a likely genotype when only the phenotype is known. Counseling, explaining parents how a child inherited a specific blood type. Let's say, for instance, explaining how two Rh positive parents can produce an Rh negative baby if both are big D and then small d carriers. We also have relationship testing, so identifying direct exclusions, such as when a child possesses an antigen that neither the parent carries. So I have here another example. So parent number 1 is homozygous O, while the parent number 2 is heterozygous A and B. So but both of the genes are considered dominant since A and B are dominant antigens. So just simply combine O and A, and then it will now become AO, and then O and B will now become BO. Therefore, the genotypes present for their offspring is AO and BO. So the percentage of their offspring having AO is 50%, then BO is also 50%. The phenotype or the blood type that could be expressed by their offspring, so we have 50% blood type A and 50% also for blood type B. I have here another example. We have both parents having AB genotypes in their red blood cells. So the genotype present in their offspring could possibly be having AA homozygous. Now we have heterozygous AB and as well as BB homozygous. So the percentage of their offspring that has AA is 1/4 or 25%, while their offspring having AB phenotype is 1/2 or 50%, and then BB 1/4 or 25%. Therefore, the phenotype that can be seen during testing, we had 25% blood type A, 50% blood type AB, and 25% blood type B. So here, you can also do this. No, so this is what I was saying, no, from the previous slide. Now let's proceed to the genes, alleles, and polymorphism. So in the study of immunohematology, understanding the relationship between genes, alleles, and polymorphism is essential for performing accurate serologic testing and ensuring patient safety. Genetic information or the genes the gene is the blueprint for the antigens we detect on the red cell membranes. The gene is like this instruction manual. So again, just to recap from the previous slide that we have discussed, a gene is the basic unit of inheritance. Biochemically speaking, it is a segment of your DNA that encodes for a specific protein, so such as a cell surface antigen or an enzyme. You can think of a gene as a single recipe in the massive instruction manual of our DNA. So these genes are located at specific fixed positions on a chromosome called a locus or a loci. So let's say for instance, the ABO gene is located on the long arm of chromosome 9. So its job is to provide instructions for the glycosyl transferase enzymes that build your A and B antigens. Now let's talk about alleles. Alleles, let's say, are the different versions of your gene. So while everyone has the same genes, let's say for instance, we all have a gene for a blood type at a specific locus or loci. So we don't all have the same alleles. Alleles are the alternate forms of a gene that can occupy that same locus. So in blood banking, alleles can interact in several ways. So alleles can be codominant, recessive, and amorphic. When we talk about codominant, both alleles are expressed equally. If you inherit an A allele from one parent and a B allele from the other, your cells will show both A and B antigens. So that's why you will be typed as group AB. So recessive, the trait is only seen if you inherit the same allele from both parents. So let's say for instance, the group O phenotype is recessive. So you must be homozygous. No, you must have the O and O, no, in order to express it. Amorphic or silent. So these alleles produce no detectable antigen product. The O gene is a classic amorph because it doesn't produce a functional transferase. So let me give you some examples. So at the KID blood group system locus on chromosome 18, you might have the JKA or the JKB alleles. So these alleles are antithetical. When you talk about antithetical, meaning they are opposite forms that can occupy the same spot on the chromosome. Now let's talk about polymorphism. Polymorphism is like a diversity of the library, at our school. So the term polymorphic describes a genetic system that has two or more possible alleles at a single locus. So if a gene only had one possible version, we would all have identical blood types. So polymorphism is what makes finding a perfect blood match a challenge. So the degree of polymorphism varies between systems. So we have also the terms highly polymorphic. Highly polymorphic, I can give you an example. The HLA system or the human leukocyte antigen system is the most polymorphic system in humans, with thousands of alleles at its various loci. So this extreme diversity is why finding a matched organ or bone marrow donor is so difficult. We also have the term lower polymorphism. So the ABO system is a polymorphic because it has A, B, and O alleles, but much less so than the RH system, which has over 50 different antigen specificities. We also have a single nucleotide polymorphism. So most blood group variations are actually the result of a single typo in the DNA sequence. Therefore, there is a change in just one nucleotide. So this tiny change is enough to create a completely different antigen. So in summary, a gene is the category, like the eye color. An allele is the specific version, like blue or brown eyes. And polymorphism is the fact that so many different versions exist across the human population. So genes are located in a specific loci on a particular chromosome. So just like what I have said the previous slide. So let's say for instance chromosome 9, it houses the ABO blood group. So the ABO blood group or the ABO gene has three different forms. So you have the A gene, the P gene, and the O gene, which are considered as alleles because they are of different versions. Again, alleles may exist for each locus. So alleles are variations of a particular gene. So a chromosome may have multiple locus or loci, but not all loci can have the ABO gene. Only one loci houses a specific gene. So let's say for instance chromosome 1 has a loci that contains the RH gene and chromosome 17 contains the Diego gene from the Diego blood group system. Allelic genes, which are antithetical, only one form of the gene could be present on a particular locus. So let's say for instance the ABO blood group has three different forms. Again, to recall, the A gene, the B gene, and the O gene, or these are known as your alleles. But only one allele can be present on a particular locus because most blood groups are having genes that are said to be antithetical. So when we talk about polymorphic genes, again, there are more than one allele that's contained in a particular loci at a particular chromosome. So another example, we have RH blood group system, so which has 5 different alleles, the big D, the big C, the small c, the big E, and then the small e. So RH antigens could be a combination of these alleles, but only 3 antigens are present. Now let's proceed to the inheritance patterns. So most blood group genes follow a codominant inheritance pattern. This is fantastic for us in the laboratory. It means that if you inherit an A gene from your mom and a B gene from your dad, both are expressed equally in your red cells. So you become group AB. In blood banking, we don't usually see one antigen bullying another. Instead, they work together so we can detect both markers simultaneously. So in most cases, blood group antigens are inherited in an autosomal codominant pattern, or the equal expression of both inherited alleles found on autosomes. So again, dominant only requires one form of the allele to express the trait. Recessive, it requires same allele from both parents to show the inherited trait. So again, codominant, if you have inherited two dominant genes, although different, both of their gene products will be expressed. Dominant, if dominant gene or double dose, or let's say homozygous, it can be expressed more. Now, if paired with a recessive gene, then it will still be expressed, but the genotype is heterozygous. Then the recessive, it is only expressed if inherited homozygously. So let me relate it in our ABO blood group system. So A and B are considered to be dominant, while the O antigen or the O is considered to be recessive. So in the presence of a dominant and a recessive gene, the dominant gene is still be expressed in its still able to express its gene product. So if you have AO and BO, still you have A and B antigens on your red cells, then you will be typed as blood group A and blood group B. But if you have received O gene from both parents, since both are recessive, they can only be expressed if it is inherited homozygously. So that's why you will be typed as blood group O. If heterozygous is paired with a dominant gene, it is always the product of the dominant gene that will be expressed. If you had inherited both the dominant genes, whether it is A or B, so homozygous for the dominant genes or heterozygous for the dominant genes, still antigens will be expressed. So in summary, dominant gene plus a recessive gene is equal to the antigen, or it will be expressed on your red cells. Now let's proceed to the term silent, amorph, and null genes. So a recessive trait can be caused by red cells expressing a null phenotype because of homozygosity of silent or amorphic gene. Silent, amorph, or null genes, these are genes that do not code for any product. So possible causes, amorphic gene inherited from both parents, or there is the action of the so-called suppressor genes. So we have examples of null phenotypes. So we have the O. O is the amorph phenotype of the ABO blood group system. Rh null, which is found in the Rh blood group, does not allow the expression of the big D, big C, small c, big E, small e antigens. We also have the Lu, A negative, B negative. So from the inheritance of both the recessive genes of the Lutheran blood group system, now the absence of the Lua antigen and the absence of Lub antigen, that's why it resulted into Lua negative, B negative. Maybe there could be the presence of a suppressor gene. So despite having dominant genes which could possibly express an antigen, but the suppressor gene inhibits the expression of these antigens. So in the study of blood bank genetics, understanding how some genes stay quiet is just as important as understanding how they are expressed. So to make this easy to visualize, if a gene is a recipe in a cookbook, an amorph is like a blank page. So the entry for that trait exists in the book, but it provides no instructions to the cell on how to make a protein. Again, to further elaborate what is an amorph or a silent gene, an amorph or a silent gene is a unit of inheritance that does not produce a detectable antigen product on the red cell membrane. While the gene is physically present on the chromosome, it is non-functional because the resulting protein is either enzymatically inactive or not produced at all. When we talk about null gene or a null phenotype occurs when an individual fails to express any antigens for a specific blood group system. So this usually happens when a person is homozygous for an amorph, meaning they inherited a blank recipe from both their mother and their father. Because there is no functional gene to produce the antigen, the red cell surface appears null or empty for that specific system. Common examples in blood banking, we have the O gene in the ABO system. So this is the most famous amorph, unlike the A and B genes which produce active enzymes to build antigens. The O gene is non-functional. It produces an inactive protein that cannot modify the precursor substance, which is why group O individuals have no A or B antigens. We also have the small h gene. The small h allele is a rare amorph. If an individual inherits two copies of the small h gene, then he or she will have a small h genotype. They cannot make the H antigen. Since the big H is the required foundation for A and B antigens, these individuals have the so-called Bombay phenotype, lacking big H, A and B antigens entirely. We also have the small s, small e, or the SE gene, the secretor status. The small SE gene is a silent allele at the secretor locus. So if you're homozygous, small SE, then you are a non-secretor and will not have a soluble A, B, or H antigens in your saliva or other body fluids. So here are the different amorph and suppressor genes in the blood groups. So again, O as the amorph gene will have the phenotype O if you are homozygous for the amorph gene. So the dominant gene for the big H would be big H, no? But if you have inherited the recessive small h gene, then there will be no big H antigens present in the red cell. Thus, it will be classified as the Bombay phenotype, which has the small h antigens in the red cells. So in your Lutheran and kid, that's why they become amorph. Maybe they contain suppressor genes. So despite having dominant genes, so the presence of suppressor genes successfully inhibits the antigens. So for Daffy, we have the double dose of the FY. So big F, small y, big F, small y gene could have the FYA negative, B negative phenotype, negative indicating the absence of the particular antigen. Now let's proceed to the Mendelian principles. So to understand blood group systems, we have to go back to Gregor Mendel. He discovered that physical traits are determined by elementen, which we now call genes. His law of independent segregation tells us that each parent passes exactly one gene for a trait to their child. Then there's the law of independent assortment, which says different traits are usually inherited separately. For example, inheriting the A blood group gene doesn't mean you'll automatically inherit a specific KL gene. So they behave independently because they are on different chromosomes. So again, to further elaborate, independent segregation, it's passing of one gene from each parent to the offspring. So there will be a transmission of a trait in a predictable fashion from one generation to the next. It illustrates that each parent has a pair of genes for a particular trait, either of that can be transmitted to the next generation. So the genes segregate and allow one gene from each parent to be passed down onto each child. While independent assortment, on the other hand, these are the blood group antigens are inherited on different chromosomes and are expressed separately. So random behavior of the genes on separate chromosomes during meiosis that can result in a mixture of genetic material in the offspring. So next, we have the different chromosomal assignments. So currently, there are almost 40 blood group systems. So most of them are considered miscellaneous, which means they are only found in a particular group of people. Whereas ABO blood group system is considered a major blood group system since 99% of the population in the world contains either A, B, or the lack of either A and B antigen. So most of the blood group systems are associated genes on their autosomes except for the XG blood group system, because the XG it is found in the X chromosome. Then we have the so-called XGA allele. If carried by the father, will be carried on all his daughters but not on his sons. So if carried by the mother, it will be passed on to all of their children. Have you ever noticed a reaction that is super strong in one tube but weak in another? So you might be seeing the dosage effect. So if a person is homozygous for an antigen like big M, so they have a double dose of that marker on their cells, often leading to a stronger reaction. If they are heterozygous, such as big M, big N, they only have a single dose and may react weakly. This is a huge clue when we are trying to identify tricky antibodies like the KID blood group system, the Duffy, or the MNSS blood group. So again, let's just define homozygous. It's the double dose of the antigen. So the genotype is made up of identical genes. While heterozygous, an individual who has inherited different alleles from each parent. Let me give you an example. No, the big A antigen, and then the O, and then the BO, and then the AB. Okay. So let's further differentiate heterozygosity and homozygosity. No, because these terms are very important in blood group antigen determination. So let's say, for instance, if you have in your phenotype JKA positive, B negative, it means that you have the presence of JKA antigens on your red cells and you do not contain the JKB antigen. However, if you are going to test for the JKA antigen, you are going to add a particular antibody, and that is the anti-JKA antibodies. Because all of the KID antigens only have JKA on the red cell, there would be numerous antibodies that would attach to the red cell, giving you a 3+ reaction. So please take note that adding anti-JKB to JKA positive, B negative will yield no reaction. So no JKB antigens for the anti-JKB antibody to attach to. Okay? Whereas if you are heterozygous to the JK antigens, JKA positive, B positive, which is found in the lower portion of our table, now you both have the JKA and the JKB antigens. Your genotype could be JKA and JKB. This means that the KID antigens on your red cell are both JKA and JKB. If you will add anti-JKA antibodies, but since it is a mixed population of your JKA and JKB antigens, it will give you a lesser heme agglutination reaction. You could see a 1+ or a 2+ reaction. Everyone, please take note that a 3+ or a 4+ grading on your agglutination reaction, it will give you a very, very strong result. While 2+ and 1+ will give you a weaker result. But still, you can see visible agglutination. Now let's proceed to the terms linkage, haplotypes, and crossing over. Sometimes genes like to travel in packages when 2 or more genes are located very close together on the same chromosome. They are linked and inherited as a single unit called a haplotype. We see this clearly in the MNSS and RH systems. For example, the big D, the big C, and the small e genes are so close that they are almost always stay they always stay together from parent to child rather than mixing and matching randomly. Some genes could interact with each other depending on whether they are inherited on the same chromosome, the cis, or on the opposite chromosome, which is the trans. So this interaction may weaken the expression of one of the antigens encoded by the genes. So in some blood groups, antigens are encoded by 2 or more genes close together on the same chromosome. and are inherited from each parent as a unit. Genes that are so close together on a chromosome that they are inherited as a unit are linked. So, independent assortment does not occur when genes are linked. So, these are called haplotypes. So again, in MNSS blood group system, M and N are alleles on one gene and S and S are on another gene. So because these genes are so close, they are inherited as haplotypes. So you can have different combinations, so big M, big S, or you can also have big M, small s, or you can also have big N, big S, or the big N and then the small s. So haplotypes occur in the population at a different frequency than would be expected if the genes were not linked. Now let's proceed to population genetics. Population genetics is the statistical study of how often specific genotypes and phenotypes occur in a given population based on the gene frequencies. So in the blood bank, this is our mathematical toolbox used to solve two major problems. Number one is finding compatible blood for patients with multiple antibodies, and number two is predicting the number of carriers for a specific blood group. Now let's talk about finding the one, or finding the compatible blood for patients. So when a patient has multiple antibodies like anti-big C and anti-big E and anti-big S, you must find a donor unit that is negative for all those antigens. So to determine how many units you will likely need to screen, you apply the product rule. So how it works, you multiply the individual frequencies of each negative trait together. So imagine a patient needs blood that is C negative. 32% of the donors, E negative 71% of the donors, and S negative 48% of the donors. So the math, 0.32 times 0.71 times 0.48 will give you 0.109. So the result, only about 11% of the population is compatible. So if you need 2 units, you might have to test 25 random bags to find them. We also have the mathematical balance, the so-called Hardy-Weinberg principle. The Hardy-Weinberg law is a formula used to determine the frequency of alleles and predict how many people are homozygous, carrying 2 of the same gene, versus heterozygous carrying 2 different genes. So we have the formulas P plus Q equals 1. This represents the total pool of alleles. P2 plus 2PQ plus Q2 equals 1. This represents the total population. So further on, I will give you more examples on the following slides. So I have here an example. So the percentages used are established antigen frequencies and can be found in the AABB technical manual. So this individual is in need of a blood. So if the patient is anemic, then the patient needs packed red blood cells. So the patient has an antibody. So the present antibodies are so the red cell that you will donate should not have the big C, big E, and the big S antigens. So if you gave a red cell that has C negative, S negative, and as well as S positive, it could lead to hemolytic transfusion reaction. That is why we need to find a blood unit whose red cells do not contain the big C, the big E, and the big S antigens. So RBC units that are negative for the big C, big E, and big S antigens would be required for transfusion. So the percentage of the donors negative for the individual antigen is expressed as a decimal point and then multiplied. So let's say for C negative, 32% or 0.32. So 100% minus 68, that will give you 32%. And then for the E negative, you have 71% or 0.71. So you're going to subtract 100% minus 29%, then that would give you 71%. For the S negative, 68% or 0.68. So 100% minus 32% would give you 68%. So based on the AABB technical manual, the portion of the population that contains the C plus antigen is 68%. So we are looking for the portion of the population that has no C antigen. So if 68% is the C plus antigen, then 32% is C negative antigen. Then E positive is 29%, E negative is 71%, S positive 32%, S negative is 68%. Next is to convert them to decimal. So C negative 32% will be equal to 0.32. E negative 71% will be 0.71, S negative 68% will be 0.68. So how are we going to solve it? C negative 0.32 multiplied by E negative 0.71 multiplied by 0.68, then that will give you 0.154 or 15%. Therefore, 15% of the population has the probability of being negative for all the three antigens. Then you are going to examine 1 out of 10 RBC units, no, which is likely to be negative for the combined antigens. Next, we have here another example. So the calculation shows that the probability of finding a unit of RBC negative for all three antigens to be 8 out of 100 units. So solving for the X shows that the antigen typing 25 units may be required to find the 2 units that are negative for all the 3 antigens in our example. So we have here, let's say for instance, remember that the patient has antibody against FYA, and then we also have the patient also has an antibody against the JKB antigen, and as well as an antibody against the big K antigen. Now, what type of red cell do we need to transfuse to the patient? So red cell to be transfused must lack, or it must be devoid of the FIA antigen, JKB antigen, and the K antigen in order to avoid any reactions. So FYA negative is 32% or 0.32. So JKB negative is 26% or 0.26. And then the big K negative is 91% or 0.91. So how are we going to solve it? 0.32 times 0.26 times 0.91, which will give us 0.076 or 8%. Therefore, 8% of the population are negative for FYA antigen, JKB antigen, and the big K antigen. So how many units should be tested for to find the 2 units of the appropriate phenotype? Then let's solve for the X. So 8 over 100 is equal to 2 over X. Then cross multiply, 8x is equal to 200. 8x over 8 is equal to 200 over 8. So you need to divide both sides by 8 to get the x. Then x would give you 25. Why do we need to solve the x? Because we need to determine how many may be required to be tested in order for us to find those 2 units of negative blood for the 3 antigens mentioned in our example. So again, x is equal to 25. Therefore, out of 100 blood bags, we need to test 25 blood bags in order to get the 2 units that are negative for FYA antigen, JKB antigen, and the big K antigen. Finally, how do we know how hard it will be to find a compatible unit for a patient with multiple antibodies? We use the Hardy-Weinberg principle. By using the Hardy-Weinberg principle formula, P2 plus 2PQ plus Q2 is equal to 1, we can calculate the frequency of a specific genotype and phenotype in a population. It's essentially our mathematical crystal ball that helps us predict how many units we need to screen to find that one perfect negative match for our patient. The concept of genetic equilibrium, so we have um, Hardy and Weinberg, which are both mathematician and a physician. So they used to estimate the frequency of genetic diseases or observe the traits. Then the formula is based on the principle that the sum of the gene frequencies, when expressed as a decimal, will be equal to 1. Okay, so this formula is used for predictions of populations at equilibrium, meaning that there is no migration, mutations, or any natural selection. So you have here the small p, which is equivalent to your allele A. We also have the small q, which is equivalent to allele small a. So if the frequency of p is 0.3, what is the value of q? So according to the formula, remember that p plus q is equal to 1. So p plus q is equal to 1. So 0.3 plus x is equal to 1. So x is equal to 1 minus 0.3. You need to transpose it in order to get the answer 0.7. So in order to check it, if your answer is correct, p plus q is equal to 1. So 0.3, which is the value of your small p, and then 0.7, which is the value of your small q, is equal to 1. So the answer, 0.7, is the value of the recessive gene. So a follow-up problem. So what proportion of the population is homozygous big A, heterozygous big A small a, and homozygous small a? So in order to answer that one, so we need the formula p squared plus 2pq plus q squared, which is equal to 1. So let's just substitute the given. So you have small p, which is equivalent to 0.3, small q, which is equivalent to 0.7, and then p times q, we have 0.3 times 0.7, which is equal to 0.21. So let's just substitute the values that we have obtained. So p squared, 0.3 raised to the power of 2 plus 2pq, so 2 times 0.21 plus q squared, which is 0.7 squared, is equal to 1. So we just have substituted the values that we have obtained, so which will give us 0.09 plus 0.42 plus 0.49, which will be equal to 1. Okay, so the answer for the homozygous big A, P2 or 0.09, for the heterozygous A small a, 2pq or 0.42, for the small a homozygous, we have q2 or 0.49. So the answer is shown like this in order to determine the proportion of the population that is homozygous dominant, homozygous recessive, and heterozygous. So this equation is usually applicable only to determine the gene frequencies in communities wherein the residents are actually permanent and no migration has occurred. So in urban areas, this is not applicable because people mostly come and go. Hardy-Weinberg formula has different applications. So it can be used in DNA testing and as well as in blood typing. So in DNA testing, making use of a polymer chain reaction, molecular studies, and molecular genetics to prove and disprove paternity. In blood typing, it is used for screening tests for paternity testing. It is not a confirmatory test. Please take note of that. So there are two types of paternity testing. So we have the direct exclusion and indirect exclusion, in which both are dependent on the obligatory gene. When we talk about the obligatory gene, it is a gene that is expected to be passed on to the offspring coming from the father. That's why it is called as a paternity testing. So maternity is always assumed since the mother is the one who's giving birth. So now let's proceed to a more in-depth discussion about paternity testing on the direct exclusion, indirect exclusion. So due to the high degree of polymorphism, blood group antigens are used in cases of disrupted paternity, although it is typically performed by molecular methods. So recognizing inheritance patterns of blood groups helps to reinforce the genetic concepts important in understanding blood groups. So the direct, again, the child has inherited a genetic marker that is not found in the mother or the alleged father. Again, obligatory gene, it is the gene that should be passed on by the father to establish probability of paternity. While the indirect exclusion, the child lacks the genetic marker that the father should have transmitted to all of his offspring. So because an indirect exclusion can be caused by a silent gene, so it is not used as the only marker to exclude paternity. Now let's go further. So we have here an example. Again, for the direct exclusion, the child has inherited a genetic marker that is not found in the mother or the alleged father. So obligatory gene, again, it is the gene that should be passed on by the father to establish probability of paternity. So in this case, in this example, the obligatory gene of the father could either be A and O. So since the child's blood type is B, the genetic marker is absent in both the mother and the father. So that is direct exclusion. Therefore, the child is not from father A since the child contains the B gene, while the father does not have the B gene. On the other hand, when we talk about indirect exclusion, again, indirect exclusion, the child lacks the genetic marker that the father should have transmitted to all of his offspring. Because an indirect exclusion can be caused by a silent gene, it is not used as the only marker to exclude paternity. So failure to find an expected marker in a child when the alleged father is apparently homozygous for the gene. So only one of the parents contains the genetic marker on the child, no, in this case. So there could be a possibility that the absence of a particular antigen or gene from the father or the expression of the antigen from the father is mainly due to the presence of the suppressor gene. So let's say for instance, so in this scenario, so we are using the KID blood group system. So the phenotype of the mother is JKA positive B negative, and then the genotype of the mother is JKA JKA. The father, on the other hand, the phenotype is JKA negative B positive. So the genotype of the father is the JKB JKB. Therefore, the obligatory gene of the father in this case is the JKB. The child now, the phenotype is JKA positive B negative. The genotype of the child is JKA JKA. So since the father is homozygous to JKB, therefore it should have been passed down to the child. But in this case, it is indirect exclusion because the absence of JKA on the father could be due to the presence of the suppressor gene that is inhibiting the expression of the JKA antigen. So it is termed as indirect exclusion because this alone cannot disprove paternity, mainly because of the possibility of the father's inheritance of the suppressor gene that has suppressed the expression of that antigen. Next, we also have the last part of our discussion, molecular genetics. So we have here different methods. We have transplantation, transfusion, hemolytic disease of the fetus and newborn, as well as donor testing and relationship testing. So kindly familiarize this table, everyone. And with that, I would like to conclude our discussion about blood banking genetics. Once again, have a good day.